So neurons communicate with each other chemically at synapses, and drugs are often acting upon the various protein machinery that is found at synaptic connections. Both, protein machines that are involved in the synthesis of specific neurotransmitters, these synthesizing enzymes, also in the storage of neurotransmitter within vesicles, right, at the axon terminal. Remember, there's specific protein machines that are called vesicular transporters that utilize ATP to grab the neurotransmitter that's synthesized in the presynaptic terminal and force it against the concentration gradient, inside the vesicle. So drugs could act there. Drugs could also act in the release of neurotransmitter into the synaptic cleft or gap, which is another critical step on, the conversion of the electrical message, the action potential, into the release of neurotransmitter, into a chemical message at a synapse. And drugs could also interfere with the removal or degradation of the neurotransmitter once it's been released into the synaptic cleft. And we're going to see that there are, all sorts of mechanisms to do this. There are, for example, presynaptic terminal transporters for some neurotransmitters that'll pull that neurotransmitter utilizing ATP, right, back inside the presynaptic terminal and remove it. And so it can no longer stimulate receptors, postsynaptically. Drugs could also interact with the various receptors, the autoreceptors in the presynaptic membrane, the heteroreceptors in the presynaptic membrane, or the postsynaptic receptors, right? Whether they be GPCRs or metabotropic receptors, or they are capable of changing, the charge difference across the membrane very quickly by opening channels within themselves, right? The ligand-gated ion channels or ionotropic receptors, right? Which they're also known as. So there are a lot that the synapse is a target-rich area, for drug action. And we're going to be talking about the various types of neurotransmitters that are, basically going to be released at, from different neurons. And there was this idea, over 100 years ago. It was called Dale's principle. And it was that there was just like each neuron had just one neurotransmitter. There was one neurotransmitter per neuron. And it turns out that Dale's principle is routinely violated. Typically there's 2, 3, even 4 neurotransmitters or chemicals that are being released, upon the arrival of an action potential, presynaptically into the synaptic cleft. And so if that's true, it turns out that even though there are usually multiple chemicals that are being released every time a neuron sort of releases neurotransmitter, most commonly there is one neurotransmitter that they call the primary neurotransmitter. It's like 80% of what is being released, or it's certainly a majority of what's being released into the synaptic cleft. So you have a primary neurotransmitter, and then you'll have maybe a secondary and a tertiary and a quaternary, depending on the relative amounts that are being released. So I want you to consider, if you have a neuron that's primarily going to release, let's say, dopamine as its neurotransmitter, we're going to see we call that a dopaminergic neuron. A dopaminergic neuron, its primary neurotransmitter, amongst the other chemicals that it may be releasing at a synapse, is going to be dopamine. But how could it, utilizing all the structural components we've been discussing that are present at a presynaptic axon terminal, how could it get more than dopamine? Dopamine and galanin, for example, or some other neurotransmitters that can be packed inside the vesicles with dopamine. What other machinery does this neuron need in order to to synthesize, to store, to release, and then to degrade more than just dopamine, but these other secondary and tertiary neurotransmitters that are packed there and released at this particular synapse as well. Well, think about it. It needs synthesizing enzymes for the other, neurotransmitters that will be released along with the dopamine. So not just the dopaminergic, synthesizing enzymes have to be expressed in this dopaminergic neuron, but other synthesizing enzymes or other, sources of the additional neurotransmitters that will be stored and then released upon the arrival of an action potential Also, think about the vesicles, right? The vesicles are where the neurotransmitter is typically stored in the axon terminal. And if you're going to have dopamine, you got to have a specialized, vesicular transporter that can pack dopamine inside, that vesicle. We're going to see it's actually what they call a monoamine vesicular transporter, because it will transport in either dopamine or norepinephrine or serotonin, any of this one class or category of neurotransmitters. But then if you're going to pack in a secondary neurotransmitter, well, you're going to need another, specialized kind of vesicular transporter that's specific for that secondary neurotransmitter to pack it inside. And then, if you're going to have, 80% dopamine and like 20% of secondary, neurotransmitter that's packed in this vesicle, well then, presumably about 80% of the vesicular transporters have to be the monoamine ones pushing in the dopamine, and only 20%, are going to be pulling in the other ones, so you get the right ratio. What's kind of fascinating is that, via these biochemical cascades that we've discussed earlier, there are changes, sort of circadian changes and changes in response to activity in terms of the expression of all these various components, at the presynaptic terminal, so that the exact ratio of like dopamine to its secondary or tertiary transmitters, that are going to be released together, might change throughout the day, or might change because of particular experiences or particular things that you're doing, actually. So there's shifts in the expression of genes, which alters, the sort of structural components that are present at the axon terminal, which changes the amount of the primary neurotransmitter in our example, dopamine, versus some of these other secondary and tertiary